Hey everybody, we're working through these cells, organelles, and structures starting on page 31 in your course pack. This information will conclude on page 34 of your course pack. So if you've heard of organs, you've probably got an idea of what organs do in your body. Organelles are similar. Organelles are simply tiny organs or sacs within a cell, and they do perform a specific job for the cell, just like organs in our body perform a specific job for our body. So these organelles are too tiny to see with the naked eye, and they serve as part of a factory, you might think, inside of the cell. So just like a factory has different people performing different functions, these organelles all perform separate functions to make that cell operate and work. Let's start off with these little tiny sacs here called ribosomes. Ribosomes can be found free, or they can be found attached to the rough ER. What I mean by being free is they may be simply in the cytoplasm, not attached to anything else. When you look at a ribosome, they look kind of like a hamburger bun. They have a large top subunit and they have a smaller bottom subunit. So just kind of like a hamburger bun, you know, if you can imagine what you see here being basically a hamburger bun. Okay, so ribosomes can be by themselves in the cytoplasm, or they may be attached to the rough endoplasmic reticulum. Here you can see them attached to the rough endoplasmic reticulum. You know, they're too small here to be able to see the top and bottom or large and small subunits. So ribosomes, we know, help to make proteins for the cell. You may already have learned that the rough endoplasmic reticulum, or rough ER, has that as its job. So the rough ER, you can see in this picture, is actually connected to the nuclear membrane. It's part of a membrane network inside the cell. Here's another view of the rough endoplasmic reticulum. It is also anchored to the nuclear envelope around the nucleus, and the smooth ER, which lacks ribosomes, is found also in that same intermembrane network. So these are part of the manufacturing portions of the cell. You may already know that the rough ER makes proteins while the smooth ER makes lipids. Once again, you can see some vesicles. These would be protein sacs that are being exported from that rough ER into the cytoplasm. And here you can see rows upon rows of rough ER, and the proteins they're making are being exported off their surface. So really a cool picture here. Kind of looks like something from another planet. Just a comparison, we call rough ER, or rough endoplasmic reticulum, that name simply because it is studded with ribosomes. Smooth ER lacks ribosomes on the surface, therefore, of course, it has this particular name. Now, beyond the difference of having or lacking ribosomes, rough ER manufactures proteins, while smooth ER makes what? It makes lipids. Now you're looking at the Golgi apparatus. It's sometimes called the Golgi body. Sometimes this is described as a stack of flattened pancakes. I think you can kind of see that, right? And these are drips of syrup coming off the edges. Well, Golgi apparatus is our packaging place. It's kind of like a UPS store or the postal service. You know, a lot of these places advertise that they will take in your items that you want to ship, they'll package them, and they'll mail them out for you. That's exactly what the Golgi apparatus does. It receives items in, perhaps proteins from the rough ER, and it allows them to be packaged into these vesicles, which are smaller sacs, and vacuoles, which are larger sacs. The Golgi apparatus prepares these vesicles and vacuoles for export from the cell. Now, the Golgi apparatus also makes a special sac that's full of enzymes. It is called the lysosome. I'll put that word down here for you. Lysosomes are specialized organelles that help to break down and recycle parts. We'll talk more about it in just a moment. Okay, so if you had to think of a summary for Golgi apparatus, I would think of it as the postal service. It receives items, packages them, and prepares them for export or for shipment. Here's a look at the Golgi apparatus, and you can see these vesicles and vacuoles being exported off their surface. Eventually, those vesicles and vacuoles will fuse with the plasma membrane and leave this particular cell. All right, so in summary, the Golgi apparatus modifies, packages, and secretes substances from the cell, such as vesicles and vacuoles. They are just simply two sizes of sacs. Vesicles are smaller sacs, while vacuoles are larger sacs. The Golgi apparatus also makes the lysosome, another organelle that we'll discuss in just a moment. All right, so in this picture you can see that we've talked so far about the rough ER over here attached to the nucleus. We've talked about the smooth ER, also attached to a membrane network that weaves from the rough ER to the nucleus. We've talked also about this packaging organelle here called the Golgi apparatus. We've talked about vesicles and vacuoles. The only difference there is the size of those. Vacuoles are larger sacs, while vesicles are smaller sacs. And of course, they may be secreted from the cell like you see here through exocytosis. That'll be our next topic in a different PowerPoint. So let's talk about lysosomes. If you've learned vocab words, then you'll know that the lyso prefix means to split or rupture. And this some prefix or suffix here means body. So it doesn't sound like a very fun place to hang out, a split body or a rupture body. Well, there's digestive enzymes inside the lysosome that helps to destroy and recycle worn out organelles and worn out cell parts. It also digests materials brought into the cell by a process that we'll discuss later called endocytosis. So maybe you know that when you are a fetus that your fingers are actually webbed together, and lysosomes help to break down and digest that skin that's between the webbing of the fingers. Lysosomes also help us to transform into a baby that's eventually born, removing things like gills in our neck area and a postanal tail. So there's a lot of transformation that must go on before babies are ready to be born. In butterflies, lysosomes also help to digest the materials that are no longer needed, so a caterpillar can transform into a butterfly. So fascinating organelle. You would be in bad shape without it, because if you don't have it, you are unable to digest and remove these worn out cell parts that can build up in your body and become toxic. Here you can see a lysosome being made from a Golgi apparatus. Right here, one is being moved off. And these lysosomes can digest or break down things that are brought into the cell. And of course, this can help us remove toxins or waste products in our bodies and help to package them up and remove them. So here you can see the lysosome engulfing a damaged organelle. Lysosomes are also important for getting rid of things like bacteria. Here you can see bacteria being brought into the cell, and the lysosomes are going to help digest that bacteria. What is spit back out over here on the far right is actually not bacteria, it's just pieces and parts. So lysosomes are critical for helping us to remove things that could potentially be harmful to our bodies. Similar is another hormone called another organelle called peroxisome. You might see the word peroxide in here. And of course there's that some suffix. It means what? It means body, which is like a sac. Okay, so you might have heard of hydrogen peroxide. Do you have some in your house? Now, if you do, you might use it for cleaning out cuts, or maybe you rinse your mouth out with it. So hydrogen peroxide can be used to help disinfect. Think of that when you remember what this particular organelle does. Peroxisomes help to break down things like alcohol, formaldehyde, and other toxins into water. You may look at hydrogen peroxide and say, hey, that looks familiar. It's actually very similar in structure to water, which is H2O. So hydrogen peroxide is part of the ingredients inside one of these peroxisomes that we're going to use to help break down these harmful toxins like alcohol into a less harmful thing like water. Obviously, water is not harmful in the right volume in your body. So, like the lysosome, the peroxisome is found freely floating in the cytoplasm. It does have a coating of membrane layer around its outside, but it is not anchored to another organelle. In this picture, you can see the peroxisome down here on the far left. It is in fact smaller than vacuoles and smaller than lysosomes, which are shown right there. You may have heard of the mitochondria before if you've taken a bio class in the past. You've probably heard it called the powerhouse of the cell. The mitochondrion is indeed a powerhouse. It helps us to make ATP. And what is ATP? ATP is energy. It's adenosine triphosphate, right? You're talking about that as a nucleic acid. And ATP is used to help us move and produce any kind of thing that requires any effort. ATP is made here in the mitochondrion. The more mitochondria you have, the more that cell is involved in making ATP. So muscle cells, for instance, would have a lot of mitochondria because they're involved in what they do, which is producing movement. Some cells will have more mitochondria than others. So you saw the previous picture of the mitochondrion. It is across those layers of membranes that we end up producing around 34 to 36 ATP molecules for every single glucose. This particular kind of cellular respiration is actually aerobic cellular respiration. So you've probably heard of aerobics before. This is a little different. This term here means with oxygen. Now, you can turn glucose into ATP without oxygen being present, but it's not a very efficient way to do it. If you're talking about gas mileage, this is good bang for your buck. For one glucose molecule, you get 34 miles per gallon or 34 to 36 ATPs. That's pretty good. Now, the waste products you make are carbon dioxide, which you exhale every time you breathe out, and water. Now, that water is not enough to live on. It's called metabolic water, but it does contribute to your overall water values for each day, but not enough, like I said, for you to live on. Where do you get the oxygen from? You get the oxygen from breathing in, and of course, the glucose comes in foods that you eat. You may recall that glucose is in fact a carb, and specifically, it's a monosaccharide. Okay, and if you remember, it is a monosaccharide because it has between 3 and 6 carbon atoms. In fact, glucose has 6, or 3 and 7 rather, it has 6. Let's talk about some proteins in your cells. These little tiny things here are called centrioles, and they're made up of hollow tubes of proteins. Here you can see that they're arranged in a particular setup. There are 9 of them around the outside of this structure, and they do provide support for things like cilia and flagella. So we'll talk about cilia and flagella in just a moment. For now, you'll want to know that they are hollow logs of protein that provide support for certain structures in your cell. Here you can see again this arrangement of triplets. There's 3 centrioles per grouping, and there's 9 of them arranged around in this tube structure here. Like I said, these centrioles do help give rise to cilia and flagella. They are also involved in mitosis, helping us pull apart and separate chromosomes. Imagine life without centrioles. You know, they're extremely important for what we do in making new cells. You can see here that centrioles are composed of very tiny structures of protein called microtubules. The types of proteins that make them up are called tubulins. You can see that word right there. So these very tiny tubes help to make these centrioles that form cilia and flagella at their base, as well as help our cells divide through mitosis. You might recognize this picture here. This is a picture of Anaphase on the left. That's a cell undergoing mitosis. You can see the chromosomes on the right there. That perhaps might even be prophase. So the way we move these chromosomes apart is through the process of using these centrioles to help build our mitotic spindle fibers or our spindle apparatus. You can see in this picture down here, this basal body is actually composed of centrioles. Those microtubules help to make those centrioles that form the base of cilia and flagella. So if you're unfamiliar with cilia and flagella, we'll talk about that in just a moment. Here's a closer look at the basal bodies of our cilia and flagella. And again, you can see the arrangement here of those tubules, those very tiny tubes of proteins. And so these basal bodies here, those are embedded in the plasma membrane or cell membrane, while you'll see this portion here stick up above. We'll talk about that, like I said, in just a moment. All right, so the term cytoskeleton should ring a bell. The skeleton of your own body helps to provide support and structure, helps you to stand upright and to move. That's very similar to what a cell skeleton does. A cytoskeleton is in fact a cell skeleton. You can see it's also made of very tiny tubes of protein called microtubules and also made up of microfilaments. So this cytoskeleton is anchored to your cell membrane or plasma membrane as well as your organelles. So it's not just that you have cytoplasm inside your cells and these organelles can squish about where they please. There's this network of cytoskeleton in there that allows us to provide support for those organelles. We also use some of these microtubules and filaments to help move along items, kind of like a transport system. So it looks very much like a web inside your cell, and that is by nature. We want to have some flexibility, but we also need a degree of support and structure, and that's what that cytoskeleton provides. Just another look here at the cytoskeleton. Not a greatly clear picture, but I think you can see the stuff in red. Here is your cytoskeleton. It does provide a framework inside the cell. Again, just a reminder that your cytoskeleton is composed of tiny tubes called microtubules and filaments called microfilaments. So you'll notice the word tubulin here that composes those microtubules and actin over here that composes these microfilaments. Hang on to that word actin. When you get later on into the muscular system, we're going to talk about microfilaments called actin and myosin a little later on. So these look like pizzas to me. What you're looking at is little tiny projections of the plasma membrane called cilia. And you're looking at these tube systems here, a 9+2 arrangement that forms the cilia. Let's take another look at cilia. This is what you're actually looking at. They have cut across cilia and they were looking down at the tube system forming them. So we weren't looking at the basal bodies, we were instead looking at the actual projections of the plasma membrane. So cilia are very important in our body. They help to create a current to move along things like mucus, and they're on stationary cells, meaning they're found on cells that don't move. Imagine your respiratory tract. There are lots of cells in the lining of your respiratory tract. It is not their job to move. However, the cilia on their surface create a current that helps to move along mucus, contaminated mucus, up to your mouth so you can cough it or spit it out. So these cilia move in a kind of a sweeping motion. Let me show you another image. Okay, you can kind of get the sense of them here. They look like French fries on the right, don't they? And here they're even smaller, but you can see they line like a shag rug those cells, especially in the respiratory tract. By contrast, flagella are on cells that are mobile. So let me reemphasize. Cilia is found on cells that are stationary, while flagella are found on cells that are mobile. So here you can see a bacterium with 5 flagella. And here you can see cells that look like sperm with only a single flagellum. So the flagellum acts like a whip to propel the cell along through a fluid environment and allows it to move or swim. All right, so not all cells are round. In fact, these are muscle cells. They're long and skinny like cylindrical rods. I don't want you to leave this lesson thinking that all cells are round and have the same sets of organelles. The organelles will vary depending on what those cells do. So some cells like these muscular cells are long, skinny like tubes. And other cells have a completely different shape. These are neurons, which look kind of like an octopus or a steamrolled squid. So they have these long extensions that extend off of their cell body, which is this region right in here. And here's an extension that comes off. So I just want you to leave this lesson thinking about the main types of organelles that we have in our cells and the variety of cells that are going to employ them. And this is something we will talk about throughout both Bio 210 and 211. Now, if you have trouble making this stuff stick, I highly recommend you take the information on pages 31 and 32 and turn them into flashcards. That way you can recognize the structure and the function readily when you see it on the upcoming test.